So we're going on here with this objective, this fourth objective in chapter 17. And we spoke about what's happening on this slide, right? We spoke about the difference between quiet breathing and active ventilation. And really the takeaway part here is that the neurons that are controlling the muscles are allowing that real expansion of the thoracic cavity, even more so for active ventilation. And that is what allows for more volumes of air to be moved in and out, okay? The next thing we're going to get into here is kind of the rhythm of breathing. Now, we know that there's a pattern to our breathing, even when we're not conscious about it, so during sleep and rest. And so we're going to speak about the things that go into generating that pattern. And full disclaimer here, guys, a lot of this is not fully understood. It's a lot, it's hypothesized or a lot of theories. And so there's a lot that we don't fully understand about how this pattern is generated, but we will go over the parts that we do kind of understand and some of the hypotheses around that. And I think it's important to point out things that we don't fully understand. One, because that's just the nature of science, right? A lot of this is evolving, a lot of the knowledge here is evolving. And then two, it may stir up your curiosity, right? One of you may go into this field to kind of study this more, which I think would be really cool. So there are two components that go into generating this pattern. One is the peripheral nervous system. So we've got things like the phrenic nerve, the intercostal nerves, both internal and external. And those are things that are outside brain and spinal cord. And then we've got some components that are inside the brain and spinal cord, so central nervous system. These are inspiratory and expiratory neurons that are located in some of the respiratory centers in the brain. And these are within the medulla oblongata and the pons. And this all kind of feeds into what's called the central pattern generator. Now, again, there's a lot that we don't quite understand about the central pattern generator, where it's located or how it behaves, but we do know that there's something else that these centers are feeding into that is then controlling the pattern, okay? So within these different respiratory centers within the medulla and the pons, we've got different types of neurons. We've got some inspiratory neurons that are usually depolarizing or firing action potentials during inspiration. And then we've got some expiratory neurons that are doing the same during expiration. We also have mixed neurons. So these have properties of both. So they're going to be active during inspiration as well as expiration. And what we see is that there are different components or different combinations of these in the different areas that we're going to speak about. All right, so the first area is the medulla oblongata, and we have two respiratory control centers here on either side. So there's two on either side. The first is the ventral respiratory group, or VRG. This is located in an area of the brain called the nucleus ambiguus. And then the other one is the dorsal respiratory group, DRG, which is located in the nucleus tractus solitarius. Now, the ventral groups, as the name implies, are a bit more anterior. They're on the lateral and anterior aspect, right? There are two of them on one on either side of the medulla oblongata. So if you remember from anatomy, right, here's the midbrain, here's the pons, here's the medulla oblongata, and at the anterior and lateral aspect on either side, we have the ventral respiratory groups. And these are usually involved in both inspiratory activity and expiratory activity. And so we can see that there are two kind of color codings here. One's in yellow, one's in blue. The yellow aspects are going to be the expiratory neurons that are involved in expiration and some parts of inspiration that we'll talk about in a bit. And then the blue areas are going to be just inspiratory neurons, so only firing during inspiration. The dorsal respiratory group, or DRG, is going to be found posterior. So the posterior lateral aspects of the medulla is where we see the DRG, and that is usually involving only inspiratory neurons, right? Mostly only inspiratory neurons. And so thinking about what these two different neurons, how they behave differently, it's thought that inspiratory neurons control the signals to inspiratory muscles. So think diaphragm, think external intercostals. In other words, we're talking about phrenic nerve communication and communication to the external intercostal nerve. Expiratory neurons are thought to control expiratory muscles, but then they're also thought to inhibit the inspiratory neurons. And that kind of makes sense because we know that expiration can be two things. It can be quiet, where it's just the relaxation of our inspiratory muscles, but then it can also be active or forceful, where we do have the inspiratory, the internal intercostals, those forced expiratory muscles activated. Okay, so the inspiratory neurons would then send communication to the phrenic nerve and the external intercostal nerve that would then send communication to the diaphragm and external intercostal, whereas the expiratory neurons can inhibit those actions as well as send instruction to the internal intercostal nerve, which would then activate the internal intercostal muscle if that is a forced state of breathing. So this is kind of how that communication would look. So we're looking at the frequency of action potentials up here, and then that is reflected by the membrane potential change down here. So during inspiration, and this is only for the inspiratory neurons. So during inspiration, what we see is the frequency of the action potentials kind of ramp up or progressively increase as we get deeper into breathing. So as you begin that breath, they're kind of more sporadic. And then as we get deeper into that inspiratory breath, the action potentials kind of speed up in their frequency. And we can see that by the increase in the curve up here. And then we see this very abrupt drop off. And I'll talk about the part of the brain that is thought to be involved in that very abrupt drop off. And then we see expiration. Okay, so during expiration, there's going to be no membrane potential change because we're now inhibiting these inspiratory neurons, which is then inhibiting the inspiratory muscles, such as the diaphragm and external intercostal. Okay, the second part of the brain that is thought to be involved here in the rhythm of breathing is the pons. And within the pons, specifically the pontine respiratory group, also sometimes known as the pneumotaxic center. I will not use this term, so you can pretty much just ignore this, but it's another term that we can think about this as. And this is thought to have different compositions. So it's got inspiratory neurons, expiratory neurons, mixed neurons. And the part of breathing that this is thought to be involved in is the regulation or the transition rather between inspiration and expiration. So if you go back here to this graph, we can see that abrupt drop off that I just talked about between inspiration and expiration. So the pontine respiratory group is thought to be involved in this transition, going from the activation of inspiratory muscles to the relaxation of those muscles, but then also possibly the activation of the expiratory muscles if it's forced breathing. Okay, and so that's kind of all we know about the pons, its location, right, in the pons itself. And then its involvement in the transition of inspiration to expiration. The third area here, the third aspect of rhythm of breathing is going to be the central pattern generator, or the CPG. Now, very little is known about the CPG. We're not really sure where it's located, although it's hypothesized to be in the medulla oblongata. And this is thought to kind of receive several different inputs. So inputs from the medulla, inputs from the pons, inputs from our frontal cortex, right? We can decide some of this. We can cognitively have some, decision-making control over some of this. And it's going to take all that information into this central pattern generator that we don't know much about in terms of its location or its mechanism. And then that is what is going to kind of feed down onto our respiratory muscles and actually control our rate of breathing. All right. So here's a more simplified model of all of that. So we've got a lot of different inputs coming in. We've got sensory inputs coming from central chemoreceptors. These are receptors in the brain that are detecting levels of acid, which is a reflection of levels of CO2. We've also got peripheral chemoreceptors. These are located in our arteries, and they're detecting, again, levels of oxygen partial pressures and levels of CO2 partial pressures. We've then got pulmonary stretch receptors that are located through the lungs. These are going to detect how much stretch is being exhibited in the lungs. Not as important in human physiology, but it does become important in some other animal species. And then we've got irritant receptors. So we've got some irritant receptors in the trachea. And be sure to make a note of this because it isn't noted in this packet, but we do have irritant receptors in the trachea that help to control things like coughing, that elicit coughing if there's any type of irritant coming through the airway. And then we've got some irritant receptors in the nasal cavity that help to elicit things like sneezing, right? So coughing receptors in the trachea, sneezing receptors in the nasal cavity, and these are sensitive to any type of airway irritant. So if you've got pet dander, if you've got pollen, any type of airway or airborne irritant would be a sensory trigger to these irritant receptors. And then the final one are proprioceptors. So proprioceptors are basically throughout our joints and muscles. They're going to be detecting the positional change of our joints and muscles. This is another reason why when we begin to exercise and we start moving those muscles and they start, changing their position, the joints change their position, that can directly input on the rate of breathing, because as we exercise more, we need to have more oxygen intake. And so that will directly feed into this pattern generator to speed up our rate of ventilation. Other inputs are the Pons, so the pontine respiratory group that we just spoke about, as well as the cortex, so the voluntary component to all of this. I can decide to take a deep breath, I can decide to hold my breath, and all of that is happening up in the prefrontal cortex. So all that gets fed into this central pattern generator, which your book is kind of saying medulla. Again, we're not entirely sure. And then that gets sent to the inspiratory neurons in those areas in the medulla that we just spoke about. So basically your DRG and your VRG, those lateral aspects of the medulla oblongata. And then that would then communicate to our nerves, our motor neurons that are going down to these skeletal muscles, which are the diaphragm, external intercostal and internal intercostal. And then they will then control the rate of our breathing. Okay, so kind of a very complex process, and thankfully so, right? This is what makes it possible for us to be able to breathe and change our rate of breathing even when we're sleeping. So if you're having a dream, and let's say in your dream you're running away from a bear, your rate of breathing is going to increase. And this is kind of the backstory of how that happens. Okay. The next thing, and probably the last thing that we'll speak about here, is the inputs to the respiratory center. So kind of getting into the details of some of those receptors that I spoke about earlier. So the chemoreceptors, the pulmonary stretch receptors, the irritant receptors, and then the muscle and joint proprioceptors. Really kind of only focusing on the chemoreceptors in terms of the most important aspect of this. So the chemoreceptors are basically detecting levels of our gases. And that I think is arguably the most important stimulus for changing the rate of our breathing. How much oxygen do we have around? How much CO2 do we have around? And so there are two types of chemoreceptors that are found throughout our body. We've got carotid bodies, so very close to the carotid sinus. We have other types of receptors, which are the peripheral chemoreceptors. And then within the brain itself, within the central nervous system, within the medulla oblongata, we have central chemoreceptors. So let's talk about the peripheral ones first. So the peripheral chemoreceptors are located at the carotid bodies. So thinking about the anatomy here, we've got the common carotids, right, coming off of the arch on the left, and then coming off of the brachiocephalic trunk. So just reviewing that anatomy here on the right. And then the common carotid is then going to bifurcate or divide into external and internal carotids. Just before it does that, right distal to where we saw the carotid sinus, we have the carotid bodies. And the carotid bodies are basically detecting the levels of these gases in the blood that's moving through these parts of the vasculature, okay? So they are not the same thing as the carotid sinus, but they're very close in proximity to the carotid sinus, if you remember those baroreceptors from the cardiovascular system. Now, these are going to then communicate to these areas of the medulla. So on afferent neurons that are going to the brain, remember afferent means going to the brain, they're going to then project their signals to the medulla oblongata, specifically to the DRG and the VRG, those two respiratory centers. And if we see different changes in our different gases, they're then going to instruct those respiratory groups to change the rate of breathing as a response to those change in gases. So this is kind of a homeostasis loop, if you think about it, right? We've got sensory receptors sending things to the integrating centers and sending responses down to muscles. Now, these receptors are very sensitive to changes in CO2. So if there's any slight change in CO2 levels, there's going to be an immediate response to change our rate of ventilation, whereas they're not as sensitive to changes in oxygen, right? There needs to be very drastic changes in oxygen before we actually see these receptors send a signal to the medulla to change our rate of breathing. Okay, so looking here at our minute ventilation and comparing that to the arterial partial pressure of oxygen, which is a reflection of how much oxygen is around, what we see is that we like for oxygen to be at 100. If we think about those partial pressures, we said that in arterial, in systemic arteries, the normal partial pressure of oxygen should be 100. And so what we're seeing here is that there has to be a very substantial dip in that partial pressure before the rate of ventilation actually increases. All right, it has to go to about 60 or lower before we actually see an increase in the rate of our ventilation, which is the response that is sent from the medulla oblongata via those respiratory centers down to our muscles in order to increase the rate of breathing. And so what this points out is that the changes to oxygen, or rather these carotid bodies and these receptors, these peripheral receptors, are not very sensitive to changes in oxygen. There has to be a drastic change in oxygen partial pressures before we see a change in minute ventilation. On the other hand, these receptors are very sensitive to CO2. And so again, thinking about where we like our arterial partial pressure of CO2 to be at 40, if there's any slight change in that, automatically there will be an increase to minute ventilation. So we like for it to be at 40. If it rises to 41, minute ventilation rises appropriately. If it rises to 50, minute ventilation will rise again appropriately, giving us this more linear curve, which is that directly proportional relationship. So any slight change in the partial pressure of CO2 results in a change in our ventilation to increase that rate of ventilation in order to blow off or to lose that CO2. And that's because our brain and our body is very, very sensitive to excess CO2. If CO2 goes greater than 90, that can result in a coma immediately. If it goes greater than 180, that can be fatal. So the body's much more sensitive to CO2 than it is to oxygen in terms of our rate of breathing. And if you think about it, CO2 we spoke about can then dissociate into bicarbonate and hydrogen ions, which are acidic. And so having excess CO2 directly results in having excess acidity, which can, denature neurons, denature proteins. It can just be a harsh environment for the brain and for other structures as well. And so the body's very sensitive to excess CO2 levels. All right. Then getting into the chemoreceptors here. So the chemoreceptors we said are located within the medulla, so within the brain. They're going to be sensitive to changes in the pH of our cerebrospinal fluid. And so they're going to be sensitive to CO2, but not directly. They're going to be indirectly sensitive to our levels of CO2. Again, thinking about this idea that carbon dioxide can dissociate into bicarbonate ions and hydrogen ions. And so those levels of hydrogen ions will affect the pH, and that is what the medulla oblongata is going to be detecting, the change in pH of the cerebrospinal fluid. Okay, and they are not at all responsive to changes in oxygen. Excuse me. So here's kind of an illustration of that. We've got the blood-brain barrier here. We've got our endothelial cells on this side. We've got the medulla oblongata on this side. And what we're seeing is that if there's excess carbon dioxide in our blood, that will cross the blood-brain barrier into the cerebrospinal fluid. That will combine with water under the influence of carbonic anhydrase, right? Remember we spoke about that enzyme. Carbonic anhydrase will then turn that into carbonic acid, which is this guy here. And then carbonic acid can dissociate into bicarbonate and hydrogen ions. The hydrogen ions, which will then change the acidity of the cerebrospinal fluid, is what the medulla oblongata chemical receptors or chemoreceptors here are detecting. So it's not directly detecting CO2 levels, but it is indirectly measuring CO2 levels, because if we have a lot of CO2, we then generate a lot of acid, and that is what is detected by these receptors.